Thank you. Okay, so I'll switch gears a little bit because I'm more on the brain side, but I'm very happy to be here and taken a lot of inputs from all of you. So I'll talk about slow oscillations and their potential role in timing. The background why I'm talking about this is that I'm coming more from the temporal domain. So I'm interested in how we learn temporal regularities and form temporal predictions to orient our attention in time. And there's two ways we can do that. One is we just orient it to certain moments, and another way is we orient it in sort of a cyclic fashion so that attention gets this rhythmic structure where we're more attentive at a certain moment or phase of the oscillation than at others. And of course, this brings up the idea that there might be a neural oscillation behind that. So a neural oscillation that's there in the brain and that sort of aligns in time with some important inputs such that we're more attentive when the inputs are most relevant. And this is called the theory of neural entrainment, which has been published by Charlie Schroeder, Peter Lakatos, and has been cited and investigated by a lot of people. So in the last years, there's been a bit of a debate what it actually means to observe entrainment. And there's two things that stand out. Entrainment in the narrow sense means that there is an endogenous physiological predisposition of the nervous system to oscillate at particular frequency ranges. In other words, it means the oscillation has already to be there, and then it tacks onto the external stimuli. So the phase and possibly the frequency of the oscillation is entrained. And so the important part here is that the periodicity comes from the neural signal. Here's just a quick primer on how we would measure this. So the idea is that we have an ongoing oscillation that phase resets to some temporally structured input. And so we can observe these peaks in the power spectrum, these narrow peaks which are taken as the signature of a neural oscillation, but we can also observe phase locking once the oscillation is entrained. So now when we come to the question, is this oscillation actually there? And I forgot to mention that here we're looking at delta oscillations mostly because they're most relevant to the types of auditory signals where we observe entrainment, which is speech or music, which typically happen in the 1 to 4 hertz range. So can we observe delta oscillations in the brain? So in invasive recordings, yes, and there's a lot of evidence from monkey studies where really in the auditory cortex we can observe these, people have observed this endogenous oscillation. There's much less evidence in humans. Here's one study with invasive recordings, ECOG, over temporal sites that found sort of this, here you see it's just the spectrum in 3D with the cortical layers added to it, but this peak here is the delta peak. And on the other hand, we have all this evidence that shows that the delta phase is instrumental in temporal predictions using both non-rhythmic and rhythmic stimuli. And here's an example from a study, like a replication study, which I did based on Gabor Stefanic's work, where you see that, so here in the two green lines, you see that the phase consistency changes with respect to the temporal expectations that we have in auditory and motor areas. And just again, to highlight this, so we have on the one hand this evidence from invasive work that there are spontaneous delta oscillations, and then from experimental studies more in humans using MEG and EEG, that there are, the delta phase plays an important role, but now we also have these periodicities in the exogenous signals, and it's really hard to actually say whether these are the same phenomena, which we do have an oscillation here. So I want to present today the work by Harish, a PhD student whom I supervised, where we went out and said, okay, let's check whether in the human MEG we can actually see those endogenous delta oscillations. And our obvious choice was to take resting state recordings, but we also took, so this was a data set that was already existing, we also took two other conditions, which were finger tapping and silent counting, with the reasonings that if participants engage in some internally rhythmic activity, it might also bring up an oscillation in their brain. So now we have these recordings from those resting states, and the first problem that we face is that, if you want to look for a delta peak, you see we are here in this part of the spectrum where there's already very high power, which is the 1/f property that we've already heard about. So the delta peaks are not obvious. And most methods in the literature that have been developed to find peaks have been validated on the alpha peaks, which kind of jump at you. But it's harder to find delta peaks. So here we use this ERASA technique to remove the 1/f from the spectrum, and then the spectra that I'll show you from now on are these so-called oscillatory power spectra where we removed the 1/f. And another remark is that here we were very careful to remove the electrocardiogram, that in the MEG we record this systematically with external electrodes, because the peak of this activity, if you do spectral analysis of the ECG, which we did here, we actually get a peak in the delta range, and this peak also shows up on the MEG sensors, which you see here. So I can tell you more about this, but just we did a very extensive ICA procedure to be sure to remove really all of the cardiac activity showing up on the sensors. Okay, now we have our sensor signal. We want to look for delta peaks, and what we found is that if we look at every sensor, there's actually quite a lot of peaks in the delta range, but with different frequencies and at different sensors. So now we needed to somehow interpret and cluster those. So we applied a k-means clustering approach, and here you see the data from an exemplary participant, where we can find 3 clusters in the delta range. That you see the spectra here, and you see the topographies here. So we did this for all of our participants, and the different conditions also. And we came up with this a little bit. So here this was a single participant. Here it's the group picture, and you see that it's quite hard to see something here. But we found that in all the conditions, basically in the counting, in the tapping, and in the resting, there was a delta peak in every subject. So the colored lines are the individual peaks, and you see that they're really quite variable. And the black lines are the average, and that you see that in the average, you don't have much of a peak anymore. So, okay, now we have these peaks. We were we wanted to convince ourselves that we're really looking at an oscillation now when we see a peak. So what we did then is we took the data from all those peaks and did another method, which from the Wojtek lab, the cycle-by-cycle method, it was also mentioned yesterday, where you take again the time domain signal and you look whether you can find endogenous periodicities in the time domain signal without computing the Fourier spectra. And what we found with this method is that only in the resting state condition, the peaks that we found, we took the sensors at which we found peaks and compared this to the sensors where we had not found peaks with the permutation approach. And we found that really in those sensors there was higher evidence of periodic activity, more bursts and longer bursts, which are first evidence that what we picked up in the peaks is actually an endogenous periodicity. So to sum this up, yes, we can observe endogenous delta oscillations non-invasively in the human MEG, but we have to go quite a long way with the signal processing, and the results are quite heterogeneous. And I didn't show these results here, but in the tapping condition, for example, we were not able to convincingly separate the oscillation from the tapping evoked response. So this really only worked in the resting state condition. And to me, this leads to the interpretation that oscillations are relatively local phenomena. So we have these populations of neurons that oscillate in synchrony, and here's a quote by Rita Hari, who wrote already in 1997 that only 1% of the cells in the cortical area of 1 centimeter determine 96.5% of the total signal. So that was based on the simulation. But I think what we have to keep in mind when we do this kind of research, as we do in humans with MEG or EEG, that oscillations might still be relatively local compared to what we pick up on our sensors. And so the future of this work would be to try and extract these oscillatory signals and see whether we can then link them to these phase codes that we observe in temporal predictions. That's it. I would like to thank the Cognition and Brain Dynamics team, in particular Harish, who did this study, Leila, who recorded the MEG data, and Virginie, who is the team leader, and you for listening. Thank you.
Thanks, Sophie, for the talk. Are there questions? Yes. I will start with the ladies.
Thanks a lot. Great talk. Very quick question. This work reminded me a little bit of the work of Thomas Andrion. Thomas Andrion in Paris. It shows that during wakefulness you can have micro episode of sleep using actually detection of this occurrence of delta oscillations. Are you aware of this, and do you think that your finding relates to this occurrence of sleep while we're awake? Or you're talking about different type of oscillations?
I would say I'm talking about different types of oscillations, but it's the resting state recordings we had here were only 2 minutes, so I don't think that people would fall asleep, but it's an important thing, and actually now we have resting state recordings of 10 minutes, where, as we heard before, it's more likely that they might be sleepy. So this Hellgren study that I cited, the delta oscillations in wakefulness can be distinguished from the ones in sleep. Like they're different regions and they look different, and also the waveform is different. So I would per se think that I'm looking at something different, but it's still important to compare directly. Yeah.
Thank you, Sophia. You probably know what I want to ask. Have you considered applying SSD? For
Sorry,
this, have you considered applying SSD?
Yeah.
Because, of course, those small oscillations, they are hidden, right? In one way of another processes. So you can think of as advanced spatial filtering, like spatial spectral decomposition, to enhance it. Have you thought or tried it?
I have thought about it. We didn't have the time in this project because the PhD time ran out. But as I said, we're recording. I think to me the biggest limitation is that the snippets we had were relatively small, and we need longer snippets, and so then we could apply more advanced methods also.
Yeah, because the idea is that these are the methods which should enhance it if there is something there. Yeah. So that's, okay.
But
just think about it.
definitely on our list. Thank you.
Okay, thank you.